right, so our next is Sammie Dolloff from Alyssa Stark's lab.
Hi everyone, my name is Sammie, and today I'm going to be presenting my thesis entitled A Balancing Act, Gait Kinematics and Behaviors of Campanose pennsylvanicus Running on Narrow Substrates. My mentor is Dr. Alyssa Stark, and all this work was done proudly in the Stark lab. Locomotion is simply the ability to move from one place to another. Animal locomotor strategies can be seen throughout the entire animal kingdom and can range from strategies including walking, running, flying, swimming, and jumping. Animals move in order to navigate their environments. They do this using the variety of strategies that I just mentioned in order to gain access to food, shelter, escape predators, and unfavorable environmental conditions. Specifically for animals moving on land, it is important that they utilize their morphology and certain behaviors in order to make contact with their substrate. And one animal that is really good at doing this are ants. Ants are everywhere, and they reside in nearly all regions of the world. In the tropics, we can find these leafcutter ants. In temperate regions, we can find fire ants, like in North America. And unfortunately, we can find ants in our kitchens. Ants make up a significant portion of the world's terrestrial biomass. In fact, 200 million tons of carbon are just reserved for terrestrial arthropods or insects, and over 50% of those biomass numbers are ants and termites. We know that ants are everywhere, but why do they matter? So within the field of biology, we can ask questions about ant morphology and their behaviors in order to answer some questions about how they're able to navigate these complex environments and forage so effectively. Additionally, ants are important ecosystem engineers, meaning that they actually shape their environment by decomposing certain trees and other debris. And additionally, ants will concentrate the nutrients that they find around their nests to create hotspots of biodiversity within their ecosystem. And outside the field of biology, using what's known as biomimicry and bioinspiration, we can actually take our knowledge and our observations from the field in order to engineer robots that can navigate their environments with the same skill and precision that ants can. Being a eusocial insect means that each ant has a different job within the colony. There is a queen who is responsible for birthing all of her daughters. There's nurses who tend to the queen's eggs. And there are workers who are responsible for nest construction and defense, and importantly for my project, foraging outside of the colony. Because worker ant morphology and behaviors are adapted to allow them to navigate so many complex environments, we can ask questions about how they are using their bodies in order to navigate these environments. Specifically, we can look at the way that they're navigating their environments, specifically on these different substrates. One parameter that we can ask questions about worker ant morphology is their kinematics. Kinematics is a branch of physics that is concerned with the forces, or is not concerned with the forces, but is concerned with the way the body is geometrically moving in order to propel a body forward, like you can see in this cheetah running here. So kinematics can be used to measure things like moving trains, running water, and moving insects and animals as well. In biomechanics, we specifically focus on kinematics because it allows us to answer questions about how animals are moving their bodies in order to navigate their worlds. In small organisms like ants, locomotor strategies and gait patterns are likely altered to reduce exposure time, which is time away from the nest, and failure, like falling off a substrate. One parameter of kinematics that my project focuses on is the hexapod gait or the tripod gait. It consists of 3 limbs, the forelimb, hind limb, and contralateral middle limb, making 3 points of contact with the surface and moving in unison in order to propel the body forward. Another aspect of kinematics that my project focuses on is duty factor. Duty factor is the percentage of the stride that the limb is in contact with the substrate. So in this graph here, we can see a run on top and a walk being demonstrated. and the duty factor is represented by these black bars here. So for a run, we see a lower duty factor because the foot is in contact with the substrate for less time, whereas in a walk, we would see a higher duty factor because the foot is in contact with the substrate for a longer length of time. Additionally, I will be measuring stride length, which is a unit of measurement between two consecutive foot contacts on the same limb. And for ants, doing this with sticky feet may be a little bit complicated. So ants have interlocking claws and adhesive pads, and the adhesive pads actually differs in size between multiple different species, and this affects how they're able to move and navigate like walking upside down. So this figure on the right actually shows the aerolium, which is the adhesive pad, being engaged when it's pulled down on a clear surface, as you can see, and here's the pad right here being engaged. Ants are using their morphologies and their sticky pads. They're able to navigate a variety of complex environments. And typically kinematics is measured on flat grounds, but for ants, they don't just walk on flat grounds. As you can see, they're moving on substrates that vary in diameter, they're varying in surface temperature and wetness. They're also walking on dirt, which is pretty interesting as well. But for my project, I am focusing on diameters. Previous findings tell us that as substrate diameter increases, ant running speed will also increase, and this is specifically in a tropical ant species. So this graph here shows the running speed on the y-axis and substrate diameter on the x-axis, and that is increasing. The running speed of the tropical ant species is increasing as substrate diameter increases. This suggests that moving on narrow substrates, ants will likely suffer a loss in performance despite making these kinematic adjustments. While this graph suggests what is going on for ants moving on narrow substrates in terms of velocity, we still have questions about why this is occurring. Why do ants slow down on narrow substrates, and how does ant kinematics change as diameter decreases? In order to answer these questions, my goals are to quantify changes in gait kinematics and speed of the Camponotus pennsylvanicus while running across dowels that vary in diameter, replicating semi-natural conditions like vines and twigs. Additionally, my goal is to quantify behavioral changes as a function of substrate diameter using behavioral parameters such as turning around, jumping, slipping, falling, and utilizing inverted locomotion. In terms of my kinematics predictions, I believe that running speed will decrease as substrate diameter decreases, as we've seen in tropical species. I believe that stride length will decrease as substrate diameter decreases, and I also believe that duty factor will increase as substrate diameter decreases. In terms of my behavioral predictions, I believe that substrate diameter will decrease with the ants performing behaviors such as jumping, falling, turning around, and inverted locomotion as that substrate diameter decreases. And this is what this graph is showing here, the total number of those behaviors decreasing as the substrate diameter increases. I also believe that ants will slip equally across all substrate diameters. And this is by virtue of the fact that when ants aren't able to engage their claws, they're more likely to slip on a smooth surface. Additionally, I believe that ants will exhibit a significant decreased kinematic performance and increased behaviors when substrate diameter is less than 2 millimeters. 2 millimeters is the average width of a specifically carpenter ant thorax. So anything less than that, I believe that they will exhibit some sort of significant decrease in kinematic performance. And the basic principles that are sort of the basis of my predictions can be observed in a person walking on a tightrope. So we have our lovely tightrope walker here. She is walking at a very slow speed on a narrow substrate. She is keeping her foot in contact with the substrate, therefore increasing her duty factor. And she's also taking really small steps. She has a small stride length. In order to do this, I collected ants from 3 different colonies using this wonderful bait of tuna and honey on trees around campus. I filmed them using a FastTech high-speed video camera at 1170 frames per second, which is pictured here. And I placed them on a glass rod here with a varying diameter, and they're filmed running horizontally in one direction. The study species that I have chosen and have alluded to are the Camponotus pennsylvanicus, the carpenter ants. I chose this species because they're found here on Villanova's campus. They're also large enough to be able to film using our filming equipment. They have large adhesive pads, which allows them to stick really well to our glass substrates. And their thorax and their gait spread is also larger than our most narrow substrates, which allows us to see the relationship that body size has while the ant is traversing these narrow substrates. These videos are analyzed using ProAnalyst software, which allows us to categorize kinematic and behavior parameters. The kinematic parameters that I'm going to be looking for are speed, stride length, and duty factor, as I've mentioned. And the behavioral parameters that I'd be looking at are jumps, slips, turnarounds, falls, and inversions. In order to highlight some of our behavioral parameters, I'd like to show you this video of a jump. So there she goes. A jump is defined as a release of all tarsi from the substrate in a directed motion. You can watch it again because it's really cool. Wow, so graceful. This is a slip and subsequent fall. So a slip is defined as an adhesive slide of the pad after it makes contact with the substrate. And a fall is just what it looks like, a release of all tarsi from a substrate in a non-directed motion. This is a screenshot of ProAnalyst, and what ProAnalyst allows me to do is to tag all 6 tarsi on the substrate using a different color. And what we do is we measure, for example, to get stride length, the kinematic parameter, we measure the distance between 1 foot as it moves across the frame. We can also get other kinematic parameters such as duty factor, and we can look at what point the foot is in contact with the substrate and when it's not in contact with the substrate. And we can also get running speed from using ProAnalyst because we are able to see at how many frames it takes essentially for the ant to move a certain distance. From our results today, I'd like to highlight some of the behavioral parameters that I have collected so far, specifically focusing on inverted locomotion across 3 different diameters and when it is used and when it's not used. This first video is filmed on an 11mm dowel, and we can see the ant not utilizing inverted locomotion, essentially just moving across very slowly on the dowel. And 11mm for an ant this size is essentially a flat surface, so we would expect this. On 5mm, we see the ant actually starting utilizing inverted locomotion and then actually moving on top of our substrate in order to complete her run. Finally, on a 1mm dowel, we see an ant, she's using solely inverted locomotion. And this is defined by an ant actually obviously moving underneath the substrate and also utilizing her pads to stick to it. And you can see right here, she's actually wrapping her tarsi around the substrate because it's narrow enough that she can walk across and do that pretty successfully. This table here shows the preliminary data that I've collected so far, mostly focusing on the behavioral parameters that I've collected. The percentages that you see are the amount of times that I have noted this behavior across different diameters. So for slipping, we see slipping occurring pretty much consistently across all diameters from 1mm to 11mm. This does coincide with my prediction that I've made earlier. And for inverted locomotion, as we've just seen, we are seeing it pretty consistently occur in 1mm and 2mm diameters. But at 5 and 7, we are seeing inverted locomotion occurring sometimes. And at 11mm diameters, we are not seeing occur at all so far. And what is really interesting about this 5 and 7mm diameter is this could be the point at which, specifically for inverted locomotion, they are utilizing one inverted locomotion or as opposed to running on top of the substrate. This could be the point yet that that this kind of changeover point occurs, but we're not sure yet. And because ant season just started this week, we are still looking at and analyzing our kinematic data. So right now I just have some of the behavioral parameters to show you. For next steps, I will complete my kinematic data collection and high-speed video analysis of the Camponotus pennsylvanicus here in the United States. I also plan on changing the substrate from a glass surface to a rough substrate in order to compare the role of tarsal adhesion on locomotion. The reason we do this is because we want to see what allowing an ant to use her claws and has on her locomotion and what behaviors she's doing. And finally, I will replicate these experiments using a tropical ant species that forage on vines. These species can be found on Barro Colorado Island in Panama. This is actually what I initially planned on doing and for my thesis. Due to the nature of my project, all data collection is strictly and season are strictly directed and isolated to the summer months. So I initially secured funding in the summer of 2020 to travel to Panama to collect locomotor data on actually Pseudomerrimax, specifically focusing on jumping locomotion. But as the story goes, that project was interrupted by COVID. So I instead spent my time looking at other forms of locomotion, specifically in my garage built by my dad. So I spent my quarantine looking at jumping locomotion in insects such as lanternflies and frogs even. And then when I was able to return back to campus, using Camponotus as a model for jumping locomotion proved pretty unsuccessful because they don't jump. So we had to morph my thesis into what you're seeing here today, into looking at this project. But I am looking forward to publishing the data that I have collected on Camponotus here in the States after I graduate, and also plan on traveling to Panama in the fall of 2022 in order to survey tropical ant species and looking at their locomotion and be able to ask questions about jumping and everything else. With that, I would like to acknowledge funding for this project was graciously donated by the Villanova Department of Biology, Villanova Undergraduate Research Fellowship, the Center for Research and Fellowship, and the National Science Foundation. I'd like to shout out all the Stark Lab members, including Michaela Desai, Mandy Kai, and Abigail Hahn for their help with data collection and method testing. I'd also like to shout out Dr. Stark. Hi, Dr. Stark, she's on Zoom, for her ever-present mentorship and leadership, and for inspiring me to pursue research beyond graduation. And my mom and dad, of course, for shaping me into the creative and curious spirit you see before you today. And with that, I will take any questions.
That was really great. I loved all of your use of gifts.
Thanks.
So I may have missed this, and I apologize if I did. How did you motivate the ants to run across the substrates in the first place?
That's a super question. Camponotus specifically do not do what they want you to do. So it's a lot of scare tactics. It's a lot of placing them specifically where you want them to be and encourage specifically their kinematics. When we're looking at behaviors specifically, we don't want to influence them to not fall off a substrate or not turn around. So how that's kind of remedied is me just placing them there, standing back with my trigger and waiting for them to do anything, and then just pressing the button on the high-speed camera as I see fit. So yeah.
Sorry, a follow-up.
Yeah.
So I saw the gif of them falling.
Mhm.
Do you place that at a certain height above the surface that makes it like impedes? I know you said you didn't want to impede them falling, but like makes it risky to fall, or like
Yeah.
is there a certain height that like
Mhm.
changes that propensity to fall?
Yeah, so these videos of the behaviors that I've collected are these specific behaviors. I don't know if it'll play. Maybe it won't. Oh, but this is, yeah, they're above a certain like distance, so they can't like sense the ground or sense anything below them. But these were specifically taken to like prompt that. So I was like on the other side scaring them and trying to get them to fall. And obviously she's on the end, so she can't go anywhere except down. So
Thank you.
So I have a question about slipping that parameter.
Mhm.
And I think you had predicted it was going to be equal across all diameters.
Yeah.
I was curious why, and because I wouldn't necessarily make that prediction.
yeah, sure. So I can actually show you slipping here occurring. So if you can kind of see here, her foot will move on the substrate and then slip a little bit after she makes contact. And this is pretty common in species with adhesive pads, because if they're not able, or for insects with adhesive pads, because if they're not able to like hook their claws onto something, they're just using their pads, and sometimes the pads get like dirty, or they just like slip by virtue of them moving their body across because they don't, move on glass substrates in the wild. So they're not really used to that. So slipping, I predict, will occur across all diameters regardless, just by virtue of them using their pads instead of their claws.
Because you would think in a wider diameter, they'd be able to avoid slipping because they'd be able to put their feet away from the edge more than on a smaller diameter.
Yeah, I think it's just by virtue of the like material of the substrate that we're using that slipping just occurs. And I also count when I look at these videos, I also count slipping like per foot I can see. So maybe not all of her feet are slipping, but one of them is slipping, and that would count towards my data. Yeah.
Nice videos, and I learned a lot about
Yeah, that's my goal, guys.
So in the thinner diameter kind of a substrate, is there a wrapping around going on there that this size is it? So they're wrapping around?
Yeah, so you can kind of see her doing that. Essentially, like, so the tarsus of the ant is like her foot, and then she's essentially wrapping like what would be like her elbow or ankle like around the substrate, which I think is really interesting. And actually, I know I mentioned that running speed I think will decrease on these substrates, but ants are pretty good at doing this. So from once I collect the kinematic data, I think that ants will, from just what I've seen, they might run it using inverted locomotion if they're really good at doing this pretty quickly. So we might see some sort of leveling off if they're able to use inverted locomotion. Yeah, Dr. String.
I have a couple questions. So first one, this might be sort of a half baked question, but how did they figure out the diameter of the substrate? And the thing that I'm asking, the antennae was really interesting.
Mm-hmm.
They like the, and so yeah, so what kind of a sensory, like how are they figuring out the diameter? And then the second question is about the kinematic analyses. Mm-hmm. Are you going to analyze it in a way in which the sequencing of those kinematic events will come out? Because I was just wondering, is it possible that there's some sequencing to those behaviors that matters? Like one change would happen first that might trigger the next change and the next change. And would you analyze it in a way that might bring that out?
Yeah, so I'll answer your first question first, but so how they sort of figure out the diameter, I don't know if you can see my tongs in here. Oh yeah, so there are my little tongs that I place them on. And what I do is I don't sort of let go of the ant until all her feet or most of her feet are placed on that diameter. And this just allows her to see, okay, I can spread my legs out, or okay, now my legs are really tiny, I only have one leg on. And then I let go once she is all the way adhered. And then from there, there's kind of like a lag in what to do. So like we can really see this in the 5 millimeter, because first she starts off using inverted locomotion, and then she realizes, oh, maybe I can walk on top because I'm a little bit bigger, because the diameter is a little bit bigger. And then your second question, in terms of analyzing kinematic data, typically we would have a different pool of ants for, if I'm understanding your question correctly, we typically have a different pool of ants per diameter. So what we just want to see is what is happening at the kinematic level and how fast they're running on this diameter, and not so much as like how the kinematic parameters are affected within each other. However, if you like, the only relationship that I would probably have is like, if I'm gathering running speed, I would also look at their stride length and also look at the duty factor of that same run. But what I really am interested in is how the kinematic parameters are affecting or are being affected by their behaviors. So I would also use those that same ant on that same 5 millimeter run to look at what behavior she's doing and how that's impacting their speed or any other kinematic parameter.
I have you were scaring them to go to the end.
Mostly.
So with the jumping, like if they, as you said earlier, they couldn't sense the ground, where
Mm-hmm.
you just leap of faith or like?
Yeah, we don't want to see the lands. They're not that good. They just kind of go, but yeah.
Okay, thanks, Sam.
Yeah.
What am I missing? Sorry about that. Okay, next is Luke Rathbun from the Obama's Eyes Lab.
Thank you. Hi, everyone. I'm Luke, and my thesis is looking at the Y6 gene family as potential tumor-associated antigens and immunotherapeutic targets in cancer. And so I've kind of done two projects here. And so my first project is looking at the Y6 gene family in mice, in which I have examined the function of Y6A in vivo using a tumor transplantation model. And my second project has to do with the Y6 gene family in humans, in which I've used the Cancer Genome Atlas to analyze Y6 gene expression in different cancers. And so some background on the Y6 gene family in mice. So this gene family has 30 members, and what they all have in common is this LU domain, which is cysteine rich. And so I've aligned a few of these members down here. So you can see these conserved cysteine residues that I've pointed out in red. And so these cysteine residues are spaced out in specific patterns to form these disulfide bridges that give each of these proteins their unique structure.